Okay, so the bioreactor group, they spent too much time on their introduction, so they were talking about very basic stuff in vertebral disc biology, structure and function, and pathology. And to be honest, all of this information, while it's useful for the student to read about, it's actually not state of the art and therefore should not be included in the state of the art report. And to give you an example, we've got the structure of the invertebral disc. This stuff's been copied and pasted from a textbook, because I know exactly which textbook it is, and even though I told the student not to include this information, she still decided to put it in the state of the art report, and she shouldn't have done, because all I did was pull out of these pages. I didn't even look at it, I didn't even mark it. To give you another example, the hydroxyapatite group also followed a similar process. They included information on the structure and function of bone, its physiology and mechanotransduction, and again, to be honest, none of that information is really relevant. They may wish to include this information in an appendix, but to be honest, it's not state of the art and it should not be included in the state of the art report. And to give you an example of what the lithotripsy group did, they wrote 2 pages on the introductory stuff on urine, on the urinary system, and on those 2 pages they've got some nice illustrations which do not look as though they've been photocopied or lifted from a textbook. Now, what you should be focusing on in the state of the art report is the critical analysis of the academic literature. So what these guys did, the bioreactor group, they went back to the literature and they read lots and lots of papers, and their heads were spinning because there was all of this information. They didn't really know how to assimilate all of this information and knowledge. And so what I suggested to the group was to try and collate this information into a series of tables. So for example, for this table, they were looking at mechanical conditioning. So they read all of these various papers, and these groups utilised different model systems, and they had utilised different mechanical conditions, and they saw various results, so the major effects, and these are all listed for each of the various papers. This is a really nice way of presenting critically the findings that other research groups have presented in their publications. To give you another example, they also looked at the types of bioreactor technologies that are available both commercially and also within research groups. And again, various research groups, they've got illustrations of the systems and the various characteristics for each of these systems, and they've described the pros and cons of each system and what actually some comments, so what could be done to overcome some of these challenges within the bioreactor systems. And what was really nice about this critical analysis was that it allowed them to identify the gap within the research findings. So what these systems did or didn't do, they could identify that gap, and then that led very nicely onto the aims and objectives. However, what this group forgot to do was actually include a page on the aims and objectives, which was actually rather very silly. I think they got a bit carried away. But if we look at the hydroxyapatite group, they discussed some of the structural characteristics of porous scaffolds, and they discussed some of the methods that can be used to look at porosity. Some of these techniques, such as micro-CT, SEM, I mean, these are methods that are used routinely by labs. For some reason, this group decided to include a description of these techniques. That is not state of the art. You know, information on how SEM works is not state of the art. What is state of the art is how research groups have utilised these methods to analyse and examine porosity and mechanical properties within various scaffolds. That's what the group should be discussing. And some of the students have included publications. They've discussed some of the findings by previous researchers, what they found, what were the pros and cons of what they found, and then found some gaps, some problems associated with these various research findings, which then led on to a page which describes the aims and objectives of the project. Compare this to the lithotripsy group. What I really liked about what these guys wrote was that it was very, very neat, it was very tidy, the information was focused, it was relevant, it was critical. They used a lot of referencing, which meant that they kept going back to the academic literature, and they were discussing what these research groups had found, what were the problems with these findings, and what was the gap. And they didn't do what the bioreactor group did, which was to include a table. Instead of including the table, what was they did? They gathered all of that information, and they were able to discuss blocks of that information, and very, very critically, which made it state of the art, which allowed the group to say, okay, this is what this lot found, but this is what we need to do now. And that's why this was a very, very good state of the art report.